Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to deal with medical biochemistry. Before I start with the topic, please like, share, and subscribe the channel. In the last session, we dealt with classification of proteins. In today's session, we will deal with quantitative estimation of proteins. First procedure, Kjeldahl's procedure. The protein sample is digested by boiling at 360 degrees centigrade with concentrated sulfuric acid in presence of copper sulfate and sodium sulfate as catalysts. The nitrogen present in the protein is reduced to ammonia, which is absorbed by acid medium to become ammonium sulfate. After cooling, the digest is made alkaline by adding excess alkali. Now ammonia is liberated, which is absorbed by a known quantity of stranded acid kept in the vessel. In this diagram, we can see that Kjeldahl's flask and Kjeldahl's trap are used to measure unknown concentration of proteins. The excess acid present in the vessel is back titrated with the stranded base, from which the liberated ammonia can be calculated. Then the quantity of nitrogen originally present in the protein is assessed. Since the proteins on an average contain 16% of nitrogen, then weight of nitrogen into 100 upon 16 or nitrogen into 6.5 will give the value of proteins in the original sample. Advantage of this procedure is it is most accurate and precise method. It is generally used for standardizing a particular protein. That protein is then used for calibrating other proteins employing other easier methods. Disadvantage is that it may take many days to get the result and is unsuitable for routine clinical work. Second method is Biuret method. Cupric ions chelate with peptide bonds or proteins in alkaline medium to produce a pink or violet color. The intensity of the color is proportional to the number of peptide bonds. The color is then compared with the standard protein solution treated with the Biuret reagent and estimated colorimetrically. In this diagram, we can see that negative Biuret test occurs because of the proteins are absent and it gives blue color, whereas positive Biuret test contains proteins are present and gives a purple color. Advantage of Biuret method is it is simple one-step process and is most widely used for plasma protein estimations. The disadvantage is that the sensitivity of the method is less and is unsuitable for estimation of proteins in milligram and microgram quantities. The third method is Lowry's method. This is based on the reduction of following CO-caltophenol reagent, that is phosphomolebidic acid and phosphotungstic acid, and by the tyrosine and tryptophan residues of protein. But the blue color is developed which is compared with that produced by a known standard. Advantage of this method is it is very sensitive and can measure protein content in microgram range. If the tyrosine and tryptophan content of the proteins of test standard vary widely, then the accuracy is lost. This is a major disadvantage of this method. The fourth one is spectrophotometric estimation. The proteins will absorb UV light at 280 nanometer. This is due to the tyrosine and tryptophan residues in the protein. Quantitation is done by comparing the absorbance of the test solution with a known standard. In this diagram, we can see that UV light is passed through protein solution, and then the light is used to measure the solution, uh, unknown protein solution. Advantage of this estimation is that it is simple, accurate, and highly sensitive up to microgram quantities. Since the color is using other chemicals is not employed, the protein is not wasted in this method. However, the instrument is costly. The next is radio immuno diffusion method or Mancini's technique. Please note that the name is radial and not radio. There is no radiation applied in this process. As the precipitation arc is moving radially outward from the point of application, the name radial is given. Here, specific antiserum is incorporated in the liquid agar and then allowed to solidify on the glass plate. Then the small vessels up to 1 millimeter diameter are cut in the agar, and the antigen, that is protein solution or patient sera, is added in the well. The plate is incubated at 4 degrees centigrade for 1 to 3 days. The antigen molecules diffuse radially around the wells and react with the specific antibody present all over the agar. In this diagram, we can see that centrally antigen is present, whereas peripherally antibody is present. The antigen moves peripherally to give antigen antibody reaction, which gives a precipitation arc at that point. A white ring A white ring is of precipitate is thin where equimolecular concentration, that is 1:1 ratio of antigen and antibody is attained. The diameter of the precipitation ring will be proportional to the log of antigen concentration. If the known standards of different concentration is included along with the test, a standard graph is plotted from which quantity of test can be obtained. Advantage of this procedure is it is simple and sensitive enough to quantitate milligram or microgram quantities of protein. Since the method is based on antibodies, the test is exquisitely specific. Since serum levels of immunoglobulins, complement components, etc., are routinely assayed by this method. The next method is nephelometry. It is based on the measurement of scattering by colloids, originally studied by Richard Zsigmondy, who was awarded Nobel Prize in 1925. Scattering of light by antigen antibody complexes was used as a method of application by Libby in 1938. Nephelometry is defined as detection of light scattered by turbid particles in solution. If albumin is to be estimated, the specific antibody against albumin is added. The resultant antigen antibody complex will form turbidity of the solution. A beam of light containing preferably laser beam is passed through the solution. If the particles in the solution will scatter light, the light turning at 30 degree to 90 degree angle, generally 60 degree, is collected and passed into a detector system. The emerging scattered light will be proportional to the turbidity of the solution and which in return will be proportional to the antigen. In this diagram, we can see that when light is passed through the solution, the light emerging at 60 degree is measured and is called nephelometry, whereas light emerging at 180 degree angle is called turbidimetry. Advantage is that this is a very rapid method and suitable for automated programs. Micrograms quantities can be accurately estimated, but the disadvantage is that the instrument and reagents are costly and need careful standardization. The next method is turbidimetry. Proteins are biological fluids like urine and CSF can be estimated by adding protein precipitation agents. The turbidity is produced in measured. The method is simple but less accurate. The nephelometry and turbidimetry are based on same principle of scattering of light by colloidal particles. In nephelometry, emergent light scattered at 60 degrees centigrade is observed, while in turbidimetry, light emerging at 180 degrees is detected. Turbidimetry is comparatively cheaper, while newer techniques are being based on immunoturbidimetry. The next procedure is radio amino assay and ELISA tests. If the proteins of nanogram or picogram quantities are to be estimated, radio immunoassay, that is RIA, or enzyme linked immunosorbent assay, or that is ELISA techniques are to be employed. Here ends the session. Kindly like, share, and subscribe the channel. Thank you.